So, good afternoon everyone. I'm Judelicia Bautista and I'm here to report the DNA analysis and I'm this criminology. So, what is the lesson objective? So, the lesson objectives explain the importance of DNA analysis and second, enumerate the steps in DNA analysis. So, lesson content. So, the lesson content is describe the DNA, its location, composition, and nature, describe the forensic DNA analysis and how it is used to solve crimes, and describe the 4 steps in DNA analysis, and describe the 4 types of DNA evidence analysis. So, the first slide is all about DNA or deoxyribonucleic acid. So, what is deoxyribonucleic acid? So, the deoxyribonucleic acid are self-replicating materials which is present near all living organisms. So, lahat ng nabubuhay sa mundo, katulad ng aso, tao, at halaman, mayroon din silang composition of DNA. And DNA is a carrier of genetic information. Second one is nature and composition of DNA. DNA is made up of molecules called nucleotides, and each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. So, saan nga ba nakalocate yung DNA? DNA location is in the cell. As you can see of the picture, the inside the cell is the DNA, chromosomes, and nucleus. So, the next slide is forensic evidence analysis and how it is used to solve crime. So, in cases where a suspect is identified, a sample of the person's DNA can be compared to evidence from the crime scene. In cases where a suspect has not yet been identified, biological evidence from the crime scene can be analyzed and compared to the offender profiles in DNA database to help identify perpetrator. So, ano nga ba yung biological evidence na tinatawag? Ang biological evidence na tinatawag is hair evidence, blood, fingerprints, urine, and saliva. That is the example of biological evidence na pwede mong makita sa crime scene rather. So, the second slide is steps in DNA analysis. So, there are 4 steps in DNA analysis: extraction, quantitation, amplification, and detection. So, what is extraction? Extraction, they extracting the DNA from the nucleus to relate the content. So, second one is to quantitation, to determine how much cell DNA was retrieved from the cell. And third is amplification, or the determination of the various in DNA test result or types from a specific region of the DNA. So, last is the detection process. DNA is separated and passed through the laser. So, what is the step in DNA analysis? The isolation of the DNA from the evidence sample containing DNA of unknown origin, and generally at a later time, the isolation of DNA from a sample from the known individual. So, the process in DNA so that the test results may obtain. The determination of the various in the DNA test result or types from the specific region of the DNA. And last is the comparison and interpretation of the test result from unknown to known sample. So, the third slide is all about the types of DNA analysis. This is the last slide. So, what are the types of DNA evidence analysis? Polymerase chain reaction, short tandem repeats, and Y chromosome, and mitochondrial DNA. So, what is the difference between the 4? So, 4 is polymerase chain reaction. And the third is short tandem. As you can see in the picture, there are the example of types of evidence analysis